Hello everyone. In this video lecture we will retrieve the coding sequence and will divide it into the codons. For this we will use the Google search engine and will write ensemble. Click on ensemble genome browser. Here you may select any of the species of your interest, and here we will write the symbol of gene of interest. And now we got the results of our search, in which I will select the GCK human gene. Here I can see its complete name, which is glucokinase, its symbols, its localization, which is on chromosome number 7, and then this gene has total 14 transcripts or splice variants, and this is the transcript table. As I told you earlier that we will prefer to select any transcript which is encoding the maximum number of amino acids. So which is the first one, which is encoding 465 amino acids. So I will select it. After clicking on this transcript, on the left side below the sequence there is the option of exons. Select the exons, and this will lead us to a page where you would be able to see the exonic sequence, the complete sequence from 5' UTR to 3' UTRs of your gene of interest just below the transcript table. One thing which I want to tell here, on the left side you can see configure this page option. When you will click on it, in my previous video lecture I showed you that whenever we want to find out the SNPs that have been reported in any particular gene, then we get the data in tabulated form, but you may also get the data labeled in the nucleotide sequence. For that purpose you will select the option configure this page, and now here there is an option show variants. You will select yes, and if you want the variants only in exons, then you will select in exons only. Suppose let's select the in exons only option, and then click on this top. On the top right corner there is a right sign. Click on it, and now you will see, refresh this page. Yes, and you will see the exonic SNPs in the nucleotide sequences. And here the variants colors are shown, like in blue color 3' UTRs and 5' UTRs available and are available, while in green color the green colored SNPs are showing those mutations which are in coding sequence. Purple, in purple color you are able to see the frame shift mutation, and red color shows the stop gain mutation. If we click on it, then you will find its location, the SNP location, its nature or consequence, like it's a deletion, and the position on the protein is 9, like it is in the 9th codon, its consequence type. There may be more than one mutation present at a single gene. In this case, you will find the two separate descriptions. The variant ID for one SNP is given here, while for the other SNP is given here. But we are just interested in retrieving the coding sequence, so we don't need these SNPs in nucleotides. So we will select no, close this page. And now we will get the sequence of the gene without nucleotides. Now we have to copy its or retrieve its coding sequence. And as I mentioned you earlier, the coding sequence always starts with the start codon, which is ATG. Now copy this part of first exon and paste it in a Word file. And in order to differentiate between the different exons, you may also color them differently. And then this is the part of exon number 2. Copy it and paste it. Then we will select exon 3, copy, paste. Next exon, copy, paste. Select all the exons one by one with great care, because if even a single nucleotide is missed, then you will be having a defective coding sequence and it will make your research faulty. So this is the point when you have to take lot of care, because if your coding sequence is fine, then your whole research will be fine and accurate. Now you can see here the last codon is UGA or TGA. Now we will color them differently just to distinguish between the different exons. After coloring them, uh, eliminate the gap between the axons. And then you will divide the whole coding sequence, as it will take too much time. So I'm just telling you what will be the next step. After joining the one exon with the other exon, with great care, you will split the whole coding sequence into the codons. And as you know, one codon comprises of 3 nucleotides. So I have some issue, sorry, with the word. In this way, you will split the whole coding sequence into codons. This will make it easy for us to read the codons and to see which codon is present at which location and which codon is encoding which amino acid. So the interpretation will be easy in future, and hence you will complete the whole coding sequence. And if you have missed even a single nucleotide while copy pasting these sequences of exons, then you will get at the end only two at the end a codon comprising of only 2 nucleotides or maybe of single, of single nucleotide.